maybe the speaker would turn on the camera. That would be
great, please.
Okay, thanks.
So we have a leftover question from Dr. Shinho's talk, which is, wait, it's here. You mentioned about the use of the research results from the lipoproteins and the lipid stuff that can be used as indicator for COVID-19 diagnosis. And then you propose that it may be used with, or developed into other platforms, which is more handy and more convenient for the diagnosis. So the question is, will it be specific enough for lipid profile?
Okay, so actually I already replied to the participants individually just now from the message. Yeah, so the animal metabolomics lipoprotein profile is very complete. So it means that the result is not just based on the LDL, HDL, but you also give a lot of information related to the subclasses. Yeah, so there are 115 parameters that can be derived directly from just one simple plasma analysis. So as a result, it's very complicated. So it means that we don't. If you are using some other technology platform, you may not have you may not able to observe this very full informations. Yeah, yeah.
All right. Thank you.
Thank you.
Okay. Okay, maybe I have another question for Dr. Sun Jae, because of looking from your model, it seems that fatty acid seems like beneficial to the host, and it seems like it just generated only from the microbe, but we don't know which one is producing it. But do you have any idea if it's the specific of gut microbial metabolite which providing the negative effect to the host, and how can we incorporate that to the models? Because I think that the reading from the recent article, there has been identify a couple more of the toxic microbial metabolite, where it's very specific to the bacteria species and also specific to the disease. I was wondering if we could we include that information to your model and then maybe make a more accurate for the predictions.
Yes, so I think in the microbiome field, most important is the functional part. That's for sure, because actually there are many functions shared between different microbiomes, but still there are some different metabolism producing metabolite produced by different species. So we need some good strategies to distinguish which species can produce that metabolite or that metabolite. I think so. The building the metabolic map, I think that's quite easy stuff. But I think most difficult one is to really track the metabolic pathway. So, for example, if we want to produce some fatty acid, we need some precursor before producing that fatty acid. So that's why I tried to make a functional cluster, was to see can we really capture the pathway level, not just single biochemical reaction. That's why I tried to make a functional cluster. So for now, the functional cluster does not really work perfectly to find what is the, for example, acetate producing pathway or propionate pathway, but still can little bit capture that fragment of the pathway. So based on good or well-developed methodology, I think we can find such information, and we can also find bacteria.
Yeah, before we move on to the other question, I still have a relevant question to my to this question, is that because I have seen recently there have been a lot of working on the meta-transcriptome and meta-proteome. Would you comment on those two that probably can contribute even more for the building up of the modeling of the human host?
Yes, I think that's a very good comment, because actually the metagenome does not have information about what kind of genes are really expressed and what is really actively producing. They try meta-transcriptome and proteome is really a very essential tool. But one difficulty is that it's very difficult to process it. That's why some people produce it, but they don't know how to analyze it. That's some challenges. But still, we need to challenge ourselves to really analyze meta-transcriptome and proteome. I think really surely think that the micro function can only be captured by meta-transcriptome, because when you see the metabolic pathway, actually like 60%, 70% of pathways are shared by different microbes. But we know that each microbe have different functions, surely. So that's why we want meta-transcriptome or proteome pipeline.
Yeah, that we also working more or less the same. We also found that we also hypothesized that using the information from the meta-transcriptome maybe it's just better than looking down from the metabolite, because most of the metabolite are unknown, and it's hard to it needs a lot of effort. On the other hand, we have rich information on the transcriptome. When it comes to meta-transcriptome, maybe it help maybe shorten the way to do this as well. Okay, thank you. So
yeah, there's a question for Dr. Manisha in the chat box. The question is about tagging proteins with GFP may affect the proteins and cell activities. So what proteins did you tag GFP with, and does it have a chance to affect the result?
So actually we didn't actually tag GFP to any particular protein, because when we generated the cell lines, we did the stable transfection of the cells with the plasmid construct, expressing GFP. So they kind of, integrated randomly in the genome of the cells. So, but we did compare the phenotype of the GFP label cells with the original cell lines, and tested them against various inhibitors, and they have the same similar IC50. So we assume that the cells maintain the same properties.
Right, thank you.
Okay. Do we still have any other questions? That's why we are working.
Not from the chat box.
All right.
So please feel free to join the discussion if you have any concerns or comments or any difficulties working on your research. So just your time.
Even the thing that you would like to share.
Yeah. Or even your pain. Yeah, so Sinja, I have a kind of concern about the integrative analysis that we usually use for integrating the omics data together, right? So most of which rely on the Spearman-Rank correlation. Would you comment on this, please?
Sorry, can you repeat the last word?
Sure. So most of which of the tools for the data omics data integration rely on the Spearman-Rank correlation. So could you comment on this point, please?
Okay. So I think this statistical thing is quite challenging in metabolism field, because the metabolism is one of the stable omics. Like for example, transcriptome we are quite easy to analyze, because for example, we compare the healthy and disease condition, actually they are really changing a lot. But in terms of metabolome, I think it's quite challenging. So we do many like PLSDA, some very brand new technique to have something, right?
Yeah.
But for now, my feeling is that even though it's slightly changing, but like for example, sometimes I see some correlation analysis even capture some certain association. But I'm not sure that what is the really best methodology. But my feeling is that this metabolomics, maybe we need some pathway analysis, not just looking at the one metabolite specifically. Maybe if we aggregate some metabolic pool, maybe that will help more. That's my feeling. So I'm trying to develop some methodology to little bit prove them and then try to do better.
That's a really good point. Thank you.
So I think if the audience has no more further questions, and also the time is right, we are about 3 minutes before the schedule, which is pretty good.
Finally.
Friday evening, such a perfect ending. So maybe you can, Dr. Jutarop, maybe you want to close this.
Me? Right, so I'll try my best. So on behalf of the organizing committee and also the Thailand Metabolomics Society, we would like to express our gratitude to all participants who show up their interest towards the metabolomics, and hopefully all of the talks today would be very beneficial for your research and probably your career in the very near future. And please come and join our society. And also we have the Facebook fan page of the Thailand Metabolomics Society, or TMS. So please follow our fan page for the upcoming events, which are going to be very amazing. And enjoy your weekend. Thank you.
Before we really end, I just had one last comment to add up. Actually, we were planning to launch the metabolomic workshop, I think in February, but however, it was unfortunate that we had the
COVID-19.
COVID-19 pandemic, second wave. So that's why we decided to have a completely online pre-conference before the grand opening of the Thailand Metabolomics Society, meaning that we're gonna have the real grand opening of the Metabolomics Society again. And initially, what we are planning to have the real, I mean the on-site and online opening. I mean what I meant for. In maybe June. And in this opening workshop, we are planning to also provide basic basic courses, and then also different aspect of application of metabolomic to the various field of biology, for example, like medical research, natural products, and environments perhaps, and then of course the medical research. And please stay tuned and follow our webpage, and then we keep feeding our news for this. Yeah, thank you very much for joining. Yes,
happy weekends.
Yeah, bye.
Bye.
Thank you.
Thank you. Have a nice weekend.
Thank you for all the speakers.
Welcome.